Welcome to Global Reach to 2022. My name is Chikezie, Onuora, from University, JUCAVM. I just want to share with us in the next few minutes part of my research experience here in JIMA. The title is Genetic Control for Yield Enhancing Trait of Onuora in entomopsis, Involanum, Lansolanum, Pempariforum under tropical monsoon climate. Yeah, high humidity has remained a major disagreeing factor to increase tomato fruit production in cold tropical climates. Most available or choice cultivars are not adapted to humid ecology. And as a result, fruit production is unable to meet up with the rapidly increasing global demand. Every year we see increase in tomato production. The demand is not being met as we have the issue of high humidity. In most places, tomato is cultivated. As you can see in the picture, you can see this is a colossal west. High humidity sometimes can be up to 100%. And that poses a challenge to farmers. While some who cannot stay with will resort to greenhouse or waiting the better season. So breeding high yield cultivars which are capable of expressing their yield potential to the fullest under humid environment is necessary. With enormous genetic variation in this wide relative Solanum pimpariforum, such as this material, the wide relative of tomato we're talking about, has the ability to establish and adapt with ease under high humidity. It also has the ability to produce very high number of fruits which are rich in nutrients and possess excellent organoleptic properties. It also has the ability to have increased fruit firmness for longer period at shelf life, has early productive process, including early fruit maturity, and so on and so forth. And several work on this combination of Lycopsychum and the Pempariforum have centered majorly on qualitatively inherited traits, such as specific disease resistance as captured by Konganga et al. 2013, fruit color by Can 212, fruit shape by Barentes 216, and organoleptic qualities like flavor, sweetness, and texture as captured by Vorips et al. 2000, and lycopene content by Kingade and Follard 2013. However, the exploitation of the variation in this wide relative for development of cultivars with improved fruit yield and tolerance or resilience to humid environment has not received attention, according to Gramazo et al. 2020. We can also recall heat tolerant, according to Eba and Chow 215, heat tolerant lines were developed through the use of the normal genetic variation in this same white Pimperiforum, and it was released by AV RODC. It is against this background that we try to check the genetic properties of this cross to know, to estimate the action of gene for tomato fruit yield enhancement, and of course check for adaptability in this humid ecology of JIMA. This work actually started with the value of some materials gathered from Nigeria, which we trained interest want to focus on brand from the materials we got from Ethiopia Research Institute and so many other places, from local farmers market and so on. The same was applicable to Nigerian materials. So we made selection two diverse materials from Ethiopia and the ones also two diverse from Nigeria. And we cross using biparental mating design, using our wide relative as a common pollen donor. This is because this prominent in those traits gene response. So we're able to raise our F1 generation, F2 crosses, and then we now calculated the gene effect. From our results, we could see that there were significant rest in all the crosses for days to flora anthesis and days to 50% flora anthesis. This implied earliness in those crosses. This is this means there's a clear-cut exertion of strength of the dominant gene for earliness from the white parent over the cultivated parent. We also notice positive significant and the negative significant additive epistasis, which shows association or dispersion of allele in parents. Like we can see some of the you can see in the cross H and the W and the TW, we can see positive additive gene effect in trait. Sorry, the pictures are blocking my view, but I think I can manage. So we recorded these positive significant and negative significant additive epistasis, which, like I said, showed association and dispersion of alleles in parents. So we also saw opposite signs of dominance and dominant gene effects in two crosses, the HW and the TW, for the same traits, days to first anthesis, days to 50% anthesis, number of aborted flowers per plant, flower length, flower width. And this suggests duplicate type of gene interaction. And when there is duplicate type of gene interaction, we try to devise means to break the dominance gene, which can be by population improvement, by biparental mating design, or by recurrent selection. By that we can break the dominance or break any linkage. Then we can concentrate genes, desired genes, and then selection can take place. The significantly positive additive gene effect found in H and W and UW for number of flower petals and total number of flower per plant in all the crosses, and also in all the crosses for number of aborted flower per plant and flower length suggests high heritability of this trait, making them fixable. The same implication would be accorded the crosses DW and TW for number of flower petals and flower per plant, although with diminishing effect due to the negative sign of the additive gene effect. Positive and significant additive dominant gene effect was found in the cross DW and UW for number of aborted flower, which suggests the importance of both additive and dominance gene effect coming from different loci. Now, improvement of these two traits, the improvement of these traits, number of aborted flower per plant, will require biparental or recurrent selection, which shall involve population enhancement, which shall help to accumulate desired gene or break unwanted linkages.
Hello, can you hear me?
Yes, I can hear you.
Yes, you are over 10 minutes already. Can you wrap up your presentation?
Okay.
Thank you. You are 11 minutes in already.
My view actually is blocked with the pictures and so on. I don't know. It's making me not to see my my traits.
That's right.
So, but then we can see the preponderance of both additive dominance and epistatic gene effect in different traits, which gives a suggestion of the fact that using a particular breeding method to improve any of these traits will be inappropriate. Now we saw the preponderance of fixable genes in most flora and fruit traits, including the total fruit yield from our experiment. Now we'll try to recommend that improvement will require simple, direct, or pure line selection, as selection intensity definitely should be pronounced at the early generation, like F2. Then also for the traits that showed non-fixable gene effects, like the dominant gene effect, were noticed in flower width, style length, style diameter, ovary diameter, which of course reveal large scope for heterosis breeding. Then also we also experienced duplicate epistasis, which control number of locus per fruit in across D and W, as well as U and W. Then also saw it in the traits days to first anthesis, days to 50% anthesis, number of aborted flowers per plant, flower length and flower width, and also W and H and W and T. This shows weak selection intensity in the early generation stage, and we'll need to, which indicates that procedure which utilize non-additive gene action, and of course able to handle non-allelic interaction such as restriction, recurrent selection, could be adopted.
Okay, thank you so much.